Hello and welcome to today's lecture for microbiology and infection prevention. Our first topic for the day is prokaryotes. We are still on the description or classification of organisms. So prokaryotes, which is also known as, or it is also bacteria, is what we are going to look at for the first part of the topic. The DNA of prokaryotes lie freely in the cytoplasm. They have no nuclear membranes. Prokaryotic cells therefore lack true nucleus. A bacteria is any prokaryote that exists as a single cell, that is unicellular, with no true nucleus. So basically put, what I'm trying to say is, when we say prokaryotes, we are talking about cells or single cells that have their DNA, or they have a nuclear material, but they do not have a membrane that surrounds the nucleus. So the genetic material that is supposed to be housed in the nucleus lies free within the cytoplasm of the cell, and bacteria falls into this category. Bacteria range in the length from 0.1 to 10 micrometer. Bacteria are said to be ubiquitous. That means they are found everywhere. Or let me put it ubiquitous. It means they are found everywhere. They can be found in the air. They can be found in water. They can be found in dust, soil, plants, animals, and that is what makes them ubiquitous. They have DNA and RNA, but they lack chlorophyll. And once they lack chlorophyll, it means that they cannot perform photosynthesis. Bacteria can be seen with compound light microscope and electron microscopes. Just like we said earlier on when we were talking about acellular organisms like viruses, we said they cannot be seen with other microscopes apart from electron microscopes. But for bacteria, its size, being 10 times bigger than a virus, permits them to be seen by other microscopes apart from the electron microscopes. What I'm trying to say is they can be seen with the electron microscope as well. The study of bacteria is known as bacteriology, just like how study of viruses is known as virology. Let's look at the structure of bacteria. If we take a bacteria into consideration, they have something we call flagella. And flagella is a whip-like structure protruding from the bacterial cell wall and are responsible for bacterial movement. So it is a whip-like, it's like a tiny structure that is long, a little bit longer, and then it protrudes out of the cell wall of the bacteria, which helps the bacteria to locomote. We also have certain structures we call pili. Pili. One is pilus, and more of them, or more than one, is called pili. Pili are hair-like projections that stabilize and attach mating bacteria during DNA transfer. So when bacteria are mating in the process of DNA transfer, they have the ability to stabilize themselves using the pili, or on surfaces, when they are on surfaces that they want to prevent themselves from being washed off or moved out of that part, they stabilize themselves by using pili. Then we have capsules. Capsules. The capsule is a slimy layer for attachment to surfaces, protection against phagocytic engulfment. So the capsule is the outermost covering of the bacteria, and it is slimy. That is, it's kind of slippery, such that it makes enemies uncomfortable to hold on to the bacteria. and it also helps them to be able to attach themselves to surfaces. Then the cell wall. The cell wall composed of peptidoglycan and prevents osmotic lysis of cell and confers rigidity and shape on cells. What I'm trying to say is that the cell wall is actually rigid. It contains something we call peptidoglycan. This peptidoglycan makes the wall thicker and firmer, which makes the bacteria it shaping the bacteria, or it gives the bacteria its shape as it appears. Then we have the cytoplasmic membrane. Cytoplasmic membrane. This encloses the cytoplasm. So within the cell wall, which is made up of the peptidoglycan, as we said earlier on, we have the cytoplasm. But the cytoplasm is first surrounded by a membrane, and this membrane is what we refer to as the cytoplasmic membrane. It composed of a phospholipid bilayer and acts as a permeability barrier for substances in and out of the cell. So this cytoplasmic membrane contains, or is made up of, phospholipids. It contains a lot of lipids and phosphorus, and what we are saying is it is semi-permeable. So it allows certain substances to enter and to move out of the bacteria. Then the cytoplasm. So the cytoplasm is a gel-like fluid containing water, enzymes, nutrients, waste products, and nuclear material. So just like in a normal human cell, the cytoplasm has gotten certain things that helps the bacteria to survive. So this cytoplasm, making up of fluid, has gotten enzymes in it. It has nutrients like protein and other substances, folic acid, that they need in order to thrive. You also have waste products which are coming from metabolic activities of the bacteria, and it has also nuclear material making up of DNA and RNA. Then ribosomes. Ribosomes are sites for protein synthesis. Ribosomes are also found within the cytoplasm, and the ribosome is where protein is produced, or protein is, let me say, in normal times, when I say protein is metabolized or cooked, let me put it that way. So that is the source where protein synthesis takes place, that is ribosomes. Then we have the vacuole, which is also found in the cytoplasm. The vacuole. The vacuole is the site for food storage and buoyancy. So in the vacuole, food is stored, and the vacuole is also, it contains air spaces that makes the bacteria to become buoyant. Like when the bacteria fall on, say, a liquid medium, it is able to suspend because the vacuole makes it air-filled and it is kind of buoyant. Then nucleoid. Nucleoid, or we have an area within the bacteria we call the nucleoid area. This area contains the genetic material, that is DNA. The nucleoid area, mostly in a normal cell, you have gotten a nucleus having a nuclear membrane and then the genetic material in it. But we said earlier on that the bacteria do not have a true nucleus. So there is no nuclear membrane, but we have a genetic material lying freely within the cytoplasm. So the area within the bacteria, that is within the cytoplasm, where the DNA is located, is what we call the nucleoid area. Then we have the plasmid. Plasmid contains supplemental genetic information. So plasmid are also organelles within the cytoplasm that also contain extra genetic material or genetic information that helps bacteria in order to give birth to their offsprings, or like to multiply. Bacterial endospore. There is a stage in bacteria that we refer to as the endospore, or the sleeping bacteria. In literal terms, we will say the bacteria is sleeping, or the sleeping bacteria. Medically or scientifically, we refer to it as an endospore. So let's look at what that means. Bacillus and Clostridium are among the few bacteria that are able to produce endospores. An endospore is a heat-resistant and non-growing structure that retain viability over long periods of time under adverse environmental conditions. So those of you who did biology, you remember organisms like amoeba. We studied amoeba and we said during unfavorable situations, the amoeba could produce a spore that would protect it. It can be in very hot environment, whatever environment. Once the environment is unfavorable, it will be in this endospore and it will never die. It will be as if it is dead, but in reality it is alive. When favorable conditions return, then the amoeba will revolve again or resurrect, let me put it that way, from its sleeping state. It is the same thing that happens in this situation that we are talking about. So a lot of bacteria can go through these processes. And I'm mentioning specific examples including the Bacillus or the Clostridium bacteria. These organisms and many other bacteria are able to produce something we call an endospore. So if they get to a situation which is not favorable, they will just produce an endospore, cover themselves, they will be there as if they are dead, but in reality they are not dead. When favorable conditions return, then they will come back to normal life and they will start to reproduce again. So an endospore have thick walls. When the environment becomes more favorable, this endospore then germinates to a vegetative cell. So like I said, the endospore has gotten very thick cell walls or very thick walls that protect the bacteria from any adverse condition. And when the environment returns or conditions returns to be favorable, then this endospore will now germinate, it will erupt, and the bacteria will become alive again. Now, let's look at the structure of the bacteria. This is the picture demonstrating the structure. We have the capsule, you can see it, we have the cell wall, we have the plasma membrane, which is the green part, we have the cytoplasm, we have the ribosomes, we have the pili, you can see the projections, we have the plasmids, which have extra genetic material, we have the nucleoid area, which is a circular, which contains the circular DNA, and then we have the bacterial flagella. You can see the long whip-like structure here. That is the flagellum. Shapes of bacteria. So the first one is cocci. Cocci means that bacteria looks round or oval in shape. Cocci that remain in pairs after dividing are called diplococci. We're talking about the bacteria multiplies through binary fission. So when the bacteria multiplies through this process into daughter cells, some of them may remain in pairs. These ones that remain in pairs, if they are the cocci type, we call them diplococci. Cocci that remain in chains after dividing are called streptococci. Examples of streptococci are Streptococcus pyogenes. Cocci that divide in multiple planes and form grape-like clusters or sheets are called staphylococci. Examples are Staphylococcus aureus. Then we have the bacillus. So we are done with the cocci, which is the round or oval shaped bacteria. We are now on the bacillus. The bacillus are rod-like or stick-shaped bacteria. So they look like a rod or a stick. Most bacilli appear as single rods. Diplobacilli appear in pairs after division, and streptobacilli appears in chains after division. Examples of streptobacilli are the anthrax bacillus. Some bacilli are so short and fat that they look like cocci and are referred to as cocobacilli. Example is Escherichia coli. Then the third category, so we looked at two, that is the cocci and the bacilli. whilst the cocci are round or oval in shape, the bacilli are rod-like or stick-like in appearance. Now we are on the third one, which is the vibro. These ones are curved or comma in shape. So they are comma shaped. Example is vibrio cholerae, the one that causes cholera. Then we have the spirilla or spirilla. This is a helical shape and are fairly inflexible or rigid body. An example of these is Helicobacter pylori, the one that causes peptic ulcer disease, Helicobacter pylori. Then we have the spirochetes. They have a helical shape and have flexible bodies. Examples is Treponema pallidum. All right. I have something here, a picture. This demonstrates the appearance. It's not very clear, but we have the cocci. You can see the round one, that is the cocci. We have the rod-like one, the rod, that is the bacilli. Then we have the curved type, that is the vibro type. Then we have the helical or the spiral type down there. The images are also here. These could be zoomed to be more visualized. You can see the coccus, that is the first one. Then we have a pair of coccus, which is diplococci. Then we have those that comes in staphylococci. They comes like a sheet-like. They are clustered together. Then we have the streptococci, those that form shape, chain-like. You can see them. All right. Then we have the bacilli type. You can see the cocobacilli, the one that looks like a rod, but at the same time also looks like oval. We have the bacillus. We have the diplobacillus. And then we have the streptobacilli. Okay, so these are just images for you to see. All right. Bacterial growth and metabolism. Bacteria grow and reproduce by binary fission. Some species grow very slowly and others grow very quickly. If I say binary fission, we'll be talking about it though. Human beings actually grow or multiply by the male fertilizing the female, and then the female will get pregnant for about 9 months, then finally give birth to 1 or 2 or sometimes 3 or 4. But bacteria actually multiply quicker or more rapidly than that. Within a very short period of time, they have multiplied to several numbers. And these ones do not pass through, or the process by which they are able to multiply like this is not a fertilizing process. We call it binary fission. And binary fission is a type of asexual cell division in which a parent cell copies its genetic material and then divides, transforming into 2 daughter cells. So the bacteria, a single bacteria, will just divide its genetic material into 2 and all other organelles into 2, and then will finally divide the cell into 2, and that will form 2 daughter cells without necessarily needing to get fertilized. This process of multiplication is called asexual multiplication or asexual reproduction, and that's all referred to as binary fission. Binary fission involves DNA replication, division site selection, DNA segregation, invagination of the cell envelope, and synthesis of the new cell wall. I'll take them one after the other so that you see what it means. So let's see the process of binary fission. So the bacterium, before binary fission, has the DNA tightly coiled. So before binary fission, we have DNA material which is in the nucleoid area of the bacteria within the cytoplasm. So these genetic material, that is the DNA, before it starts to go through binary fission, it is tight together, or it is tightly coiled together. The parent cell grows to its maximum size. So during the process of binary fission, the parent cell will grow, it will elongate on itself to the highest or the longest size it can be. The cell duplicates its chromosome, a process called DNA replication, so that two exact copies of the genetic material exists inside the parent cell. So this is a much more complex process, but what it does is that the DNA will replicate, or it will duplicate, it will divide into two, and it will be having the same genetic material in either side of the two genetic or two divisions. The DNA is pulled to separate poles of the bacterium as it increases size to prepare to splitting, that is segregation. So the DNA that has been separated, one half will move to one pole, that is one end. You know, the bacteria has become elongated. So one of the DNA, that is the new DNA that has been formed, will move to one pole of the bacteria, then the other one will also move to the other pole of the bacteria. New cell wall begins to grow and to separate the bacterium. Now, the organelles within the bacteria, half will be moved to one side, another half will be moved to one side, and then the cell wall will start to invaginate in the middle. It will start to separate in the middle into two equal halves. That is what we refer to as the invagination. The new cell wall fully develops, resulting in the complete split of the bacterium into two daughter cells. So I'll show you images to demonstrate what I'm saying. But the cell wall has now divided into two. We have one daughter cell here and we have one daughter cell there that have gotten genetic material in either of them. The new daughter cells have cell walls, tightly called DNA, ribosomes, and plasmids within them. So look at these images and then you are able to tell exactly what I'm saying. So this is the binary fission in a prokaryote or a bacteria. You can see the first one, that is the number one. You have only one cell and you have genetic material in the middle or one side of it, and you have other organelles. Your ribosomes and your plasmids, they are all inside. Now, in the second cell, you can see, or the second stage, you can see that the genetic material has started elongating. Meanwhile, in the first cell, what is the first stage, the genetic material was tightly coiled in the middle, or tightly coiled within the cytoplasm. Now, in the second stage, the genetic material has started elongating. In the third stage, the genetic material has separated into two. Then, in the fourth stage, you can see that the genetic material is still separated into two, and the cell wall has started invaginating. That is, it has started cutting in between the middle or trying to divide into two. In the fifth stage, you can see that there is total separation, or there is separation of the cell into two. You have genetic materials in here and you have genetic materials in there, and including other cellular organelles or cytoplasmic organelles. Now, in the final stage, which is the sieve stage, you can see that there is total separation into two daughter cells that have gotten identical genetic materials in them. So this process is what we call binary fission. Some bacteria go through primitive form of sexual reproduction called conjugation. What we're saying here is, even though majority of bacteria multiply through binary fission, we still have some bacteria that go through a primitive form of, or a basic form of sexual reproduction called conjugation. Bacteria which require oxygen to survive are called aerobic bacteria. So there are certain bacteria that cannot survive without oxygen. These bacteria are referred to as aerobic bacteria. Bacteria which die immediately in the presence of oxygen are called anaerobic bacteria or anaerobes. So anaerobic bacteria are those that cannot survive in the presence of oxygen. Some bacteria eat simple sugars, while others break down complex sugars. Hydrocarbons or environmental pollutants. Let's look at phases of bacterial growth. Phases of bacterial growth. Bacterial growth goes through the following stages. We have the lag phase. What I'm trying to demonstrate here or talk about here is, let's say one single bacteria enters into the body. This single bacteria will not be able to cause any harm, but it has to multiply into dozens. That is, it has to go through possibly binary fission or conjugation to be able to multiply to numbers before they will be able to start fighting against the host cells. So if a bacteria enters into the body, it goes through the following stages. So let's see, the first one is the lag phase. Lag phase. The lag phase, during the lag phase, that is when the bacteria begin to adapt to the new environment. So adaptation to environment, new enzymes are produced. So let's say E. coli has gotten the opportunity to enter into your urethra. It enters in there, and over there it is a new environment to it. It will first go through the lag phase, where it begins to adapt itself to the new environment, and then it starts to produce enzymes to make it adapt to the system. That is during the lag phase. Then we move to the log phase. The log phase is when maximum growth rate is attained. During the log phase, the bacteria have started multiplying and has multiplied to a very high level. So at this stage, they are multiplying in very high numbers. That is during the log phase. Then we have the stationary phase. That is balance in division and death of bacteria, hence growth rate is 0. At this point, the body might have sensed that there is a foreign agent or there is a bacteria or an antigen in the system, and the body has started producing antibodies against it. So there is now a fight between the organisms and then the antibodies, or maybe the person has started taking some antibiotics and it has started killing the bacteria. At this point, though the bacteria is still multiplying, it is still being killed. It is being killed as well, either by antibodies or by the medicine. So as a result, if it is 100 or 1,000 bacteria that is produced in a day, about 1,000 of them are also killed by the antibodies or the medicine. As a result of these, there is no increment in the growth rate. At this stage, we call it the stationary phase. Then we have the phase of decline or the decline phase. That is when the antibodies might have overcome the bacteria and the bacteria is dying at a very high rate. So we now have a decline in the rate of multiplication of bacteria. That is during the decline phase. All right. So that is what the phases of bacterial growth looks like. Then let's look at culture media. Culture media. Culture media, as we call it in microbiology, is a medium which is solid, semi-solid, or liquid preparation of nutrients for the growth of microorganisms. So we want to grow organisms like, say, bacteria, maybe for an experiment or something. In doing so, we develop a medium that will be suitable or comfortable for their growth. So this medium could be solid, it could be semi-solid, or it could even be liquid. Anything at all that will be suitable for the bacteria, which maybe we have added some nutrients, proteins or sugar or glucose or something to make the bacteria comfortable to grow. This type of medium is what we refer to as a culture media. Now, what is a culture? A culture is a collection of microbial cells in a solid or liquid medium. So we have a medium. Once you have a medium, if you've been able to put bacterial cells or spores in this medium and they start to grow, we now have a culture. So a culture is the medium that is housing growth of bacteria cells. A pure culture, what is a pure culture? A pure culture is made up of only one type of microorganisms. So you could have a medium that is having different organisms growing at the same time, like E. coli, like Salmonella typhi, plus Streptococcus aureus. But in a situation whereby you have a medium that is having only one type of bacteria growing, we call that one a pure culture. Alright, so generally if you say culturing, what at all are we referring to? Culturing involves placing a sample of specimen on or in a culture medium. This medium has nutrients that allow the microorganisms, such as bacteria and fungi, to grow. The medium is placed in an incubator at 37 degrees Celsius for proper growth. Colonies of microorganisms are seen as they multiply. So when we say culturing, what we are doing is we have developed a medium, and then we go to pick spores or seeds of bacteria, or cells of bacteria. We come and grow them like how we just plant. Maybe you want to nurse some tomato seeds. Then you look for soil, and then you come and you make sure the soil is suitable for the development or the growth of tomato seeds. Then you come and put the tomato seeds in it. You start to water it, and then within a few days, the tomato seed starts to sprout out or germinate. That, when we do such for bacteria, we call it culturing. Environmental requirements of microbial growth. So what is required for proper microbial growth or microorganisms to grow? We need adequate nutrients such as nitrogen, vitamins, phosphates, sodium, calcium. All these are needed in order to ensure the proper growth of bacteria. Temperature is also needed for the growth of certain bacteria. These bacteria falls in these categories. We have the psychrophiles, we have the mesophiles, and we have the thermophiles. Some of them actually need very cold temperature, whereas others need very high temperature. Like the psychrophiles needs a very cold area to be able to grow, whereas the thermophiles needs a very hot environment to grow in. Then we have osmotic pressure. There are organisms that need a level of pressure or liquid pressure to be able to grow. These organisms include the halophils, the osmophils, and then the xerophils. So if you take like xerophils like these, they need an environment that is totally without water to be able to grow, whereas the halophils actually needs a water medium to be able to grow in there. Then we have pH. Some organisms actually need a pH to some level to be able to grow. You know, pH is either acidic or alkaline. So if you take organisms like acidophils, they need a much more acidic environment to be able to grow, whereas alkalophils needs an environment that is much more alkaline. Okay. Then we have oxygen. Some organisms actually need oxygen to be able to grow. We have some called obligate aerobes. These ones, obligate aerobes, means that without oxygen they cannot grow. So they absolutely need the presence of oxygen to be able to grow. Then we have the facultative anaerobes. Facultative anaerobes. These ones can grow in an environment where there is no oxygen, whereas they can also grow in an environment where there is oxygen. So if there is oxygen, they can grow. And if there is no oxygen too, they can maneuver themselves in a way so that they can still grow in there. Then we have the obligate anaerobes. The obligate anaerobes are organisms that need an environment where there is absolutely no oxygen to be able to grow. If there is oxygen, they cannot survive at all. Okay, so these are the requirements of microorganisms for survival or growth. Now, our next topic will be naming of bacteria. Naming of bacteria. The Linus binomial system is what has been used or based on for the naming of bacteria. And this system involves the genus and species names, which is used in the naming process. Example, we have a bacteria called Streptococcus pneumoniae. The first part of the binomial name is the genus, and this one is written with a capitalized first letter. So the first letter of this part is capitalized. So you can see the Streptococcus, the first letter, which is the S, is capitalized. That is the genus. So the second letter is the species name, and it is written in lowercase letters. I'm talking about the second word, not the second letter. The second word, which is the pneumonia, is the name that represents the species of the organism. And it is mostly, or it is supposed to be written in lowercase letters. Remember, the first one, which is the genus name, was written with the first letter being capitalized. Now, the binomial name is written in italics or underlined. So you can either italize it or you underlined the two words. So every organism using this binomial system has gotten two names. The first name stands for the genus, that is the genus from where the organism is coming from. And the second name stands for the species from where the organism is coming from. And the first name, always remember, which is representing the genus, has its first letter capitalized. Whereas the second name, which represents, or the second word, which represents the species from where the organism is coming from, has all lowercase letters. This name is written in an italic form or it is underlined. And like I said, the example given here represents a binomial naming procedure. That is Streptococcus pneumoniae. All right, so let's proceed. Now, classification of bacteria. We are classifying bacteria in this topic. And I'm saying that classification of the bacteria could be based on the following patterns, either by the morphology, the staining reaction, the growth and nutritional characteristics, the cell wall constituents, or the genetic characteristics of the bacteria. So let's look at normal flora. If we say normal flora, what are we talking about? Normal flora, they are benign bacteria and fungi that are permanent residents of certain body parts like the skin, colon, nose, mouth, and the vagina for protection. Normal flora are commensals but become opportunistic parasites if conditions change. If I say benign bacteria, I'm referring to bacteria that are there which may not necessarily be causing any harm to us. It may not be causing any harm to the body part where they colonize, but they may rather be benefiting that body part, or they may be serving as a dual purpose, driving benefit and also benefiting the part where they are colonizing. And I say normal flora can be commensals, but they actually become opportunistics if the conditions change. If I say commensals, I mean they can be there without necessarily causing any harm, but rather protecting the human part of the body, or they can just be there without doing anything to the body but enjoying or benefiting from the body. But then, when the conditions change, they may become opportunistic. Opportunistic means they will start to cause a disease on that part of the body. Example, if you take Staphylococcus aureus, which colonizes the skin. In fact, Staphylococcus aureus is normal flora on the skin. They are on the skin and they serve as normal flora on the skin. If you take Streptococcus viridans, they colonize the respiratory tract. They are normal flora within the respiratory tract. If you take Escherichia coli, they are normal flora within the gastrointestinal tract. If you take Candida albicans, they are normal flora within the genitourinary system. But like I said, if the situation, for instance, let's go to the skin. If you take Staphylococcus aureus into consideration, they are normal flora on the skin. They protect the skin, they prevent pathogenic organisms from colonizing and causing skin diseases. But let's say there is an abrasion on the skin and the normal flora come into contact with the dermis. Now, they are not comfortable with the dermis, or the environment has changed, the situation has changed. Since they do not know the dermis, they are going to cause infection to the dermis. So in this case, they have become opportunistic. All right, let's look at Rickettsia or Rickettsia. Rickettsia are rod-shaped or spherical bacteria that live in the cells of arthropods. They live in the cells of arthropods. Arthropods includes organisms like ticks, lice, fleas, and mites. They contain both DNA and RNA and reproduce by binary fission. So Rickettsia contain both DNA and RNA, and they are able to reproduce by binary fission. Rickettsia are obligatory intracellular parasites. If I say they are obligatory intracellular parasites, I mean they can only survive or thrive within the cell. Outside of the cell, they do not behave as living organisms, or they cannot thrive. They do not form spores and have no flagella. So unlike other bacteria, that is, other bacteria, that's the prokaryotes, that have the ability to form spores, and they also have flagella. Rickettsia do not have spores, they cannot form spores, and they do not also have flagella. They are gram-negative, and they are stained by dyes like Giemsa and Machinvello. They are also rudimentary forms of bacteria. That is, they are very basic or very simple forms of bacteria. Rickettsia can be grown using cell culture, experimental animals, and embryonated eggs, which means we can use this one as culture media to grow Rickettsia. Rickettsia diseases are transmitted through bites and waste products of the arthropods. So if a flea or a mite or a tick bites you, you stand a chance of suffering condition that is as a result of Rickettsia. Diseases include endemic typhoid. This one, it is a lice. When a lice bites you, or you come into contact with waste products of the lice, you can get infected, and then you will develop the condition called endemic typhoid. We also have the Rocky Mountain spotted fever, which is gotten from the tick, and then we have the scrap fever as well. Let's look at chlamydia. Chlamydia. They are also rudimentary, so just like the Rickettsia, they are also very basic form of bacteria, and they are obligate intracellular parasites. If I say obligate intracellular parasites, I've already explained this. They cannot survive or metabolize or live as normal organisms outside of the cell. They have both DNA and RNA, and they also multiply by binary fission. Chlamydia are gram-negative and stained by dyes like Castanida and Machiavello. So these organisms are gram-negative organisms, so they are not gram-positive, and they are stained by the following dyes, Castanida and Machiavello. They are isolated in cell culture as well. So when you do cell culture, you can actually get chlamydia out of them. Chlamydial infections include trachoma, ophthalmia neonatorium, cervicitis, non-gonococcal urethritis, respiratory tract infections, etc., etc. So these are the commonest types of infections or diseases that is as a result of chlamydia. Let's look at mycoplasmas. Mycoplasmas. These are rudimentary form of bacteria, so they are also a very basic form of bacteria. Mycoplasmas are facultative anaerobic bacteria. If I say facultative anaerobic bacteria, it means they can actually survive in oxygenated environment, and they can also manage to survive in an unoxygenated environment. They do not have particular shape, that is, they are pleomorphic. So mycoplasmas do not have a specific shape, like say they are cocci or they are bacillus, or no. they are what we call pleomorphic organisms. They have a cell membrane and they also have both DNA and RNA. They have no cell wall and are said to look like fungi because they have filaments. Mycoplasmas have no flagella like other bacteria does. Examples of mycoplasma are Mycoplasma pneumoniae, which causes atypical pneumonia. Now let's look at spirochetes. Spirochetes are long, helical, and slender bacteria. They are too thin to be visualized using routine microscopy, but they can be viewed using dark field and electron microscope. If I say dark field, it is a form of microscopy whereby the background is darkened and then these throw more light onto the organism to get reflected to the lens of the microscope. Or we can use a much more advanced microscope called the electron microscope. These can help to visualize spirochetes. Spirochetes have internal flagella called axial filaments, which are embedded in the cell wall of the bacterium. Syphilis and Lyme diseases are all caused by spirochetes. So, this brings us to the end of this session. Thank you very much.